Hi everyone, this is a virtual lab for AP Biology Investigation 5, which covers photosynthesis and uses the floating leaf disc protocols to encourage students to explore photosynthesis in the lab. So the photosynthesis overview shown here, reactants to the left, water, carbon dioxide, and light are required in this series of reactions, and the products are carbohydrates, oxygen, and water. So just as an overview for the experiment, the reactants represent possible independent variables for the experiment, whereas the products represent dependent variables for the experiment. So we'll use one of these three products as a readout, a dependent variable for our experiment. So you will need a few things from around the lab: baking soda, soap, a light source, hole puncher, syringe, some sort of leaves. We will use spinach, cups, and markers to set this experiment up. The first thing you want to do is make a bicarbonate solution. So we will make 300 milliliters of 0.2% bicarbonate solution. Quick math application question: how many grams of baking soda would you add to do that? So, and also as in the way of an overview, we're adding this bicarbonate as a source of carbon for photosynthesis in this solution. Let's label some experimental cups for our first experiment. So the first experiment is going to be a control experiment, a proof of principle experiment, if you will. So one cup is going to have carbon in it, and the other will not. So let's add 3 centimeters of bicarbonate solution to one cup and label it with CO2. And then the second cup will be just water labeled without CO2. The next thing we need to do is make a soap solution. So we're going to dilute 5 milliliters of dish soap into 250 milliliters of water, and we will use very small amounts of that dilute soap solution in other cups. So we'll take that dilute soap, add it into our experimental cups, 1 or 2 drops at the most. We want to be careful not to mix it up too rigorously. We do not want bubbles in our cup while the experiment is being performed. The soap helps the solution to infiltrate the leaf discs. So you'll notice in a minute we need to sink these discs to start the experiment. And the way that we do that is we put it into a syringe, and the soap helps that solution to get into the discs. Next, we'll have to punch some leaf discs. So use a hole puncher to take a spinach leaf and punch out 10 or more. We're going to need 10 sunken discs for our experiment, but I always encourage students to punch out a few more in case something happens to a few of the discs in the degassing process. So use that hole puncher, avoid the veins running throughout the leaf. So you can see here I've punched it just to the right of the main vein in this spinach leaf, and there's what we'll be working with, the little discs. Okay, so be careful when handling these discs. They are sensitive to too much handling. We're going to put the discs that we cut out into the syringe. So I like to cut the discs over a piece of white paper so I can see where the discs are. And we're going to basically take out the plunger from the syringe, use the piece of paper to add the discs to the empty barrel of the syringe, then replace the plunger, pushing it down towards the tip of the syringe, being careful not to smash any of the discs as we do that. So you can see that here we have our discs. We've opened up our syringe. We then, once we get the discs into the syringe, we slowly push that barrel down. We will then draw up some solution from our experimental cups, and then you will remove all air from the syringe in order to start the degassing process. So how to degas the spinach discs. You're going to put a cap on the tip of the syringe. So a lot of these syringes come with little plastic caps. I've also found just putting your thumb over the hole very useful. But you put your cap on or your thumb over the hole and you pull on the plunger. Okay, so this creates a negative pressure, ultimately creating a vacuum that forces the gases out of the discs. Okay, once the gases have been removed from the discs, they will sink to the bottom of the syringe like those shown here, and the experiment is ready to start. So we're going to take those degassed discs and add them to our experimental cup. You have to do this rather carefully. It takes a little bit of finesse to remove the barrel from the syringe and to quickly dump the discs into your cup. This is a successful run where all of them stayed at the bottom of the cup. Sometimes you'll see some floaters. Remove those from the experiment and make sure that you have 10 fully sunken discs at the bottom of your experimental cup. Going back to this overview slide, we can see here that we're supplying all the necessary reactants for photosynthesis in this experimental setup. So we have water, the cup is full of water. We have carbon dioxide in the form of bicarbonate dissolved in the water, and we're going to provide a light source. Okay, any of those can be manipulated in the lab experimentally, which we'll show in a moment. In our first experiment where we have carbon dioxide in one cup and we do not have it in the other. And these create opportunities for independent variables. The one that we will use is oxygen, which I'll explain in a moment. So how does this experiment work? Well, you have to recall a little bit of the anatomy of the leaf to see what's going on and how we take advantage of photosynthesis to create this experimental setup. So recall, if you take a cross-section of a leaf, the green portion that makes up the majority of the center of the leaf is the mesophyll layer. So the mesophyll layer is full of mesophyll cells that have lots of chloroplasts, and the chloroplasts have lots of thylakoids that perform photosynthesis. So we have taken all of the air out of the leaf discs by degassing them. So we've essentially started a clock where once these cells start to perform photosynthesis, they will once again create oxygen. So as oxygen begins to accumulate again in these air spaces, the gas will encourage the discs to now float because they're full of oxygen gas. Okay, so we take advantage of that in the experiment. So our dependent variable is rising of discs, which is an indirect measurement of oxygen production in photosynthesis. Okay, so once we have our discs sunken, we put them under a light source and start the experiment. Now, a quick reminder about setting up a nice controlled experiment. The only thing that we want to change in the experiment is the dependent variable. The independent variable. So the independent variable is what we're manipulating. Everything else needs to be held constant. That is, for this experiment, if we have one cup over here that has bicarbonate as a form, for CO2 in the experiment, you also have a cup over here that does not have it. So the independent variable that we're looking at is the presence of CO2. So everything else we need to keep constant. So in other words, these lights need to be the same wattage, they should be the same bulb type, the cup should be the same distance. The only thing that differs is this has bicarb solution. This does not. This has just water. So we're looking at the role of carbon dioxide in making these leaves float. You could easily hypothesize that these will not float because it doesn't have CO2, whereas these would float because they do have CO2. They also have water, both of them, and they both have a light source. So we're just looking at this from a closer view. This is the side view of the cup. So again, for experiment 1, where we're setting up controls, you have a light source, you have water. Inside the water, you have dissolved bicarbonate solution and your sunken discs. Okay, so in this way, this experiment is a positive control. That is, we know that this experiment is going to work. It is going to be a predictable positive outcome because we know that if you have bicarbonate solution, water, and light, that photosynthesis will occur and these discs will begin to float. You'll see that experimental data in a moment. So that's a positive control. We will see this work. Whereas over here, this is a negative control because we're relatively sure that this is not going to work because it's lacking bicarbonate. It does not have the CO2 required for the experiment. So while we can say this is an experiment studying the effects of CO2 on photosynthesis, we already know that you need CO2 for photosynthesis. So for me, for my students, I'm basically showing you that this is a positive control and a negative control for this experiment. So we'll perform this experiment for 15 minutes. I'd like for you to stop and create your own data table to track the movement of 10 discs over 15 minutes. Okay, the experimental data that follows is in 1-minute intervals. Okay, so for experiment 1, this proof of principle lab, you could also say that we're studying CO2 in photosynthesis. This is the result that you get in the first minute. So now we're looking down at the cups from a topside view. So this would be the surface of the water, the top of the cups looking down. And from this data at this time point, we can see that in the cup with bicarbonate at minute 1, there are no discs floating, whereas in the without bicarbonate solution over here in this cup, you can also see that there also no discs floating in the first minute. So let's see what happens over the course of 15-minute experiment with a cup that has bicarbonate and a cup that does not have bicarbonate. Here's your result at the end of the second minute, end of the third minute. Make sure that you're jotting this down in your data table. End of the fourth minute, end of the fifth minute, end of the sixth minute, end of the seventh minute, end of the eighth minute, end of the ninth minute, end of the 10th minute, end of the 11th minute, end of the 12th minute, end of the 13th minute, end of the 14th minute, end of the 15th minute. So now your experiment is concluded. Take a look at your data table, make sure that you've filled it out properly. And the results for this experiment were rather predictable, but we're going to practice constructing graphs to set up experiment 2 and 3. So go ahead and finalize your data. Here's how I'd like for you to show your data. So you know, the tendency is going to be, well, let's just graph the number of discs that have risen over time, and that's acceptable for experiment 1. That's pretty straightforward, given the data is not very exciting. But ultimately, I'd like you to determine the ET50. That's the estimated time taken for 50% of the discs in the experiment to rise. Okay, so that ET50 is an important number for us. It's even better when expressed as a rate, so you can express the estimated time for 50% of the discs to rise as 1 over ET50, and that's what we'll do for experiment 2 and 3. I will go over this in a little more detail in class. So now I want you to create the experimental cups for experiment 2, in which we're studying light intensity. Okay, so for cup 1, we will put 3 centimeters of bicarbonate solution, and for cup 2, we will put the same solution, because now we're not studying the effects of bicarbonate on photosynthesis, so that is going to be a constant for us in this controlled experiment. What we are interested in studying as an independent variable is light intensity. So for our control, since we know that 100 watts of fluorescent light bulbs does the trick for photosynthesis based on our previous experiments, I am interested in seeing experimentally, if I manipulate the wattage of the bulb and increase the energy in that bulb, can I increase the rate of photosynthesis? I think that's a logical experiment. I think it's logical to predict that you would increase photosynthesis based on increasing the wattage of the bulb, though experimentally you never know. So let's do the experiment. This is what the experiment looks like in this diagram. I'm going to ask you to make a diagram like this for experiment 3 down the road when you do your own inquiry lab. But basically what we're looking at here is the study of light intensity on photosynthesis. So here are some of the constants. You have bicarbonate solution for the liquid, bicarbonate solution for the liquid. So both cups have water, both cups have a source of CO2. Here's how the experiments vary. So our positive control, we used the 100 watt fluorescent bulb in our previous experiment and showed that it would that photosynthesis would occur. So this is going to be our positive control for the experiment. We are interested in studying the effects of increasing the wattage on photosynthesis. Will that increase the rate of photosynthesis? So this is what we're actively manipulating. In this case, we have a 120 watt bulb to study. Here's your experimental data for the second experiment. At minute 1, you see no floaters in either cup. Minute 2, still no floaters. At the end of minute 3, at the end of the 4th minute, at the end of the 5th minute, at the end of the 6th minute, at the end of the 7th minute, at the end of the 8th minute, at the end of the 9th minute, at the end of the 10th minute, at the end of the 11th minute. You can pause this. Obviously, I'm going a little quickly through here, but pause it and record your data. End of the 12th minute, end of the 13th minute, end of the 14th minute, and end of the 15th minute. So now you're done with experiment 2. Make sure your data table is filled out so that you can construct your graph that shows the rate based on estimated time of 50% of discs. For experiment 3, I'm going to ask you to create your own experiment. So knowing what you know about photosynthesis, what would be an interesting independent variable to study? What would you like to manipulate in an experimental setting to see how it impacts photosynthesis? Okay, draw a picture of your experiment similar to the ones in this video, and make sure you clearly define what your independent variable is, what your dependent variable is, identify 2 constants in the experiment, and identify clearly what your control is. You'll see this on a Google Doc in class, but again, we're going to include graphs. I always like for you to practice graphing. I'd like for you to hypothesize and draw conclusions for each experiment, some of which I've already mentioned in this video. There will be some questions on the Google Doc, and then I'd like to see your inquiry lab drawing with the variables identified. Okay, so I hope this has been interesting for you. We'll see how it goes.